Okay, fourth lecture in the cancer theme. So far we've talked about cells, we've talked about the different types of cells, we've talked about the parts that are found in cells, what it means to be alive, and that one of the defining characters of living cells, well, of living things, is that they're made of cells, and that all cells contain DNA, they all contain ribosomes. So this really important idea is that your genetic material, your genes, are there in order to provide the blueprint or the instructions for making proteins, and proteins really make the cell work and alive and dynamic. Last lecture 3, we talked about cell division, how cells are born, they die, they divide in order to make new cells to grow or repair. And we start to introduce the idea that really cancer is a breakdown of this process. Okay, it's an irresponsible, it is an inappropriate division of cells. Okay, so we said DNA and mutation of DNA is always at the heart of cancer. Right? So cancer is a genetic disease. It's always linked to some change in DNA. So now we're going to kind of bridge the gap. We're going to talk about how mutations in DNA can be related to the cell cycle and therefore related to cancer. Okay. So we're building, all these lectures build on the previous lecture. Okay, so you really have to understand the content, especially of lectures 2 and 3, to make sense of this fourth lecture. So here's a note slide right out of the third lecture. Okay, this should look familiar to you. It's just again the kind of the life of a cell. The cell is born here, goes through this cycle, and then at this point a new cell is born. Okay. Here we have the cell, it grows, it makes new organelles. In this time of its life, the cell is copying its DNA. Okay, and we know that it's copied its DNA because we see 2 copies over here where there was once one. Grow some more, right? And then at this point, we take those copies through that very orderly process called mitosis. Prophase, metaphase, anaphase, telophase, where we take those 2 copies and we split them, put them into piles, make new nuclei, split the cell in 2, right? And there we go. We've made our new daughter cell. Isn't it beautiful? Okay, so the new thing we're going to layer onto this, okay, we're having, if this doesn't make sense to you, hit the pause button, go back, look at mitosis. Gain an understanding of the big picture and of the details. Otherwise, it's pointless to go on. Right? You can sit here and watch the next 40 minutes, but it's totally pointless. Right? So, assuming we understand what's going on in the cell cycle, let's add on a new component here. Okay. Now, it looks a little different. We see it is a cycle. We see the arrows. It's a circle. In this case, it's flipped upside down so that the mitosis phase is at the top, but largely it's the same thing, right? So cytokinesis would happen at the end of M phase here, and we would give birth to a new cell. Then we have the G, growth phase, S, synthesis phase, and then another growth phase before we then split the nuclear material during mitosis. What we see here, the new thing, are these little reddish orange bars that appear on the cell cycle. And what this is it's called the cell cycle control system. Okay, and as the cell proceeds through its life cycle, there are places, just like when you're in a math test, you stop and check your work. Okay. It's like if you're trying to go into a club or a bar, there's going to be somebody that stops and checks your ID. They want to make sure that you're supposed to be there. right? This cell cycle checkpoint, cell cycle control system, right? It stops the cell cycle at these very specific points, checks for specific things before the cell is allowed to proceed to the next part of the life cycle. And let's look at these examples. So we'll start, right, new cell is born, it's growing, it's adding cytoplasm, it's getting more organelles, right? It has, so, and then before it proceeds to the S phase, the cell stops, and there are protein molecules that do all these jobs. Checks to make sure, are you getting big enough? Do you have enough nutrients to, because it's a very energetic process to make new copy of the DNA. It's an energetic process to grow, make new organelles. So do you have enough food? Do you have enough nutrients? Are there growth factors there? And we're going to check to make sure that the DNA in all the chromosomes found in the nucleus of the cell are whole and undamaged. Okay? If all these things are true, we check the DNA, we make sure we've got enough provisions in terms of nutrients, we're big enough. Yes, then we proceed to the S phase. In the S phase, again, we're going to make a copy of all of the chromosomes. Okay, we're building those sister chromatids. We go into the G2 phase, right? Grow some more. Before we split the copied chromosomes, there's another checkpoint. And the checkpoint is going to, again, make sure the cell's big enough, but it's going to make sure that during the S phase, the DNA was replicated faithfully. It wants to make sure that the copies are exact and that they are good copies. Okay? If the replication happened in an orderly fashion, then the cell is allowed to go through that checkpoint, right? And then mitosis happens. Chromosomes condense, nucleus disappears, spindle apparatus comes, attaches onto the chromatids during prophase, right? Lines them up in the middle of the cell. But now before we proceed to anaphase and split them apart, there's a spindle assembly checkpoint. Okay, we're going to stop. We're going to make sure that the microtubules from the spindle apparatus have attached to the centromere of the chromatids. Okay, if everybody's attached, because think about it, if this side attaches to the chromosome, the chromatids, and this one doesn't, right? When anaphase happens and the spindle apparatus pulls, instead of splitting two copies, we're going to take both copies down to this end. That's going to result in an extra chromosome. And we've seen, right, if you look at things like Down syndrome, that is caused by this type of separation problem. Okay, so those checkpoints make sure that in the end, what's the goal in mitosis was to produce two cells that are exactly the same as the parent cell. Okay, and so we see at all three checkpoints, at spindle assembly, G1 and G2 checkpoints, what's one thing that is consistently checked up on is, are the chromosomes attached? Is the DNA damaged? Was the DNA replicated properly? Right? Because we do not want to make a new cell that contains wonky directions, right? We don't want to make a daughter cell that has the wrong codon, and which will add the wrong amino acid, which will make a weirdly shaped protein, because that's how diseases are caused. Okay, so that is the cell cycle control system, and this we're building up. We're getting close to cancer. Okay, here's the same cell cycle control system. Here's those G1S, G2 phases, and mitosis in the middle. Okay, the new thing here is that, of course, proteins are directly related to this cell cycle control system. How does the cell know to proceed through a checkpoint? How does the cell right? These are just molecules inside of your cells. There's no little people down there that are checking everything out and push the button and the toll booth comes open, right? The way that the cell knows to proceed, or actually not to proceed, is at the direction of protein growth factors. So this growth factor, we see this little upside down yellow triangle, is a type of protein. If that protein is there in the right shape, it will tell the cell to go through the G1 checkpoint and to proceed into synthesis, right? And actually, this is really an oversimplification because there's a whole bunch of proteins involved here. And if any one of these proteins is damaged, we can affect the cell cycle control system. And when you affect the cell cycle control system, you affect how quickly a cell divides, right? And I'm hoping now, if you put a little thought into this and paid attention to these very exciting lectures, then you're going to say, okay, well, obviously that's why we're talking about this with cancer. If a cell divides too much, the cell can be on its way to developing cancer. Okay, so then healthy cells become cancer cells when this system breaks down. Okay, when the cell cycle control system goes haywire. That's when we start to, our cells, cancer cells don't come from the outside. Cancer cells are our cells that have changed and mutated. Okay? Here is a duct. Say it's salivary duct in your mouth. Okay? The lumen is just an empty space surrounded by living cells. Here's a bunch of cells. You can see the nucleus in each of the cells, right? Now, normally, well, say you burn your mouth or something happens where one of the cells dies. How are we going to replace that cell? Mitosis, right? Boom, cytokinesis, we replace that cell. Normally, a cell, if it's surrounded, it's fine, it's healthy, it's surrounded by other healthy normal cells. That cell has what's called a contact inhibition, which will tell the cell, okay, it would not be appropriate for you to divide right now. There's no need for you to divide. Just keep doing your job, right, and don't worry about dividing. But when mutations happen and that cell cycle control system starts to break down and the cell's not getting the signal that it's not appropriate to divide right now, what forms is called a tumor, rather. Okay, so a tumor is just shown here in the pinkish type cells. It is a mass of cells that's not supposed to be there, right? This is supposed to look like the one on the left. The middle, the weird thing is all these cells, and they start to block ducts. They block cancer. Tumors can block blood vessels, cause all kind of problems within the body. Okay, but this tumor at this point is not considered cancerous. It's considered benign, a benign tumor. Okay, so it is what it's written here. Excessive proliferation means, production or making more of something. And so there's too much proliferation, but yet the cells remain in one single mass. Okay. Now, further mutations that affect the cell cycle control system, and again, well, this is a new idea, is that it's not just one mutation that causes cancer. It's not that you have a cell in your lung and then you have a cigarette and then immediately there's one single base in that DNA that changes and you have full-blown cancer. Cancer is a progressive disease that's caused by multiple mutations. Okay? So what we get here is we see on the far right a malignant tumor. And malignant tumors, the difference between malignant and benign is that malignant tumors don't stay put. They actually start to burn through their surrounding tissue and it can invade neighboring tissue and start growing tumors there too. Okay, and so we have the same kind of light pink color right? Say maybe this cell was the original cell to mutate. Say this cell here mutates. When it divides, what does it produce? It copies that mutated DNA and it makes mutated cells, right? And then say this cell with one mutation gains another mutation, right? And that becomes like this reddish colored cell here. And then when the reddish colored cell goes through the S phase and makes a copy of its DNA, it's going to copy the already mutated DNA. And if it has a second mutation, it passes that along too, okay? And it makes these doubly mutated cells. I think that they say the average is maybe like up to 8 mutations will cause a malignant tumor to form. And now we have cancer, okay? And cancer is, that's one of the big reasons that cancer is so dangerous, because it is malignant and it moves throughout the body. Okay, we'll talk more about that. Okay, so cancer is caused by mutation, right? But it's not just any part of a chromosome or any one part of your genome that mutate. There are some mutations that count a lot more than others when it comes to the formation of cancer, okay? And we have this family of genes, right? A gene, remember, is just a code for a protein. It is one segment of one chromosome. There's a whole bunch of these genes called oncogenes, right? And these are the ones that generally have to be mutated in order to produce cancer. And in fact, look in the name of the oncogene. Onco means cancer. So oncologist is a cancer doctor. Oncogene is a cancer gene. So mutations count more when these things called proto-oncogene, right? Proto-oncogene, it's not an oncogene, it's a prototype of before oncogene. It's a potentially cancerous gene. It's normal and we need them there, okay? But if something goes wrong with that proto-oncogene, that can then become, if it becomes mutated, it can become an oncogene, okay? So how do they go from proto-onco to oncogene, right? Some mutation, right? Viruses can actually insert DNA into your chromosomes. And sometimes the virus will insert an oncogene, pretty much causing a tumor, okay? Or the mutagens that we talked about last time, expose yourself to too much alcohol in your stomach lining. A proto-oncogene, a normal healthy gene, can be mutated and become an oncogene, okay? So let's look a little bit more about these proto-oncogenes and why do we have them? Why is that a normal healthy cell if it can potentially become a gene that tells the cell to basically invade and attack your own body? What is a proto-oncogene? Well, proto-oncogenes, all a gene ever does is code for a protein. Remember, the gene doesn't make the protein. The ribosome makes the protein. The role of the gene is to tell the ribosome the sequence of amino acids in that protein, therefore how it's going to fold up and what its job is going to be. So the proto-oncogene is the recipe for this type of protein that is called a growth factor. And just like the name implies, what does this protein do? Remember, we go back to our previous slide here. There's that growth factor. Why is that growth factor there? Because the ribosome got the code from a proto-oncogene. And when that growth factor is there, that cell will divide, okay? But we can see, right, I hope that you can see when something goes wrong with this growth factor, that's going to lead to uncontrolled cell growth, right? And that's what cancer is. So the conversion from proto-onco to oncogene is basically a mutation. So let's look at a couple of examples. I find, for me, I can read this stuff over, but I really need to see a diagram and try to spend some time understanding the information in that diagram. But we're gonna see 3 mutations, and in all cases, the change in the DNA could potentially lead to the development of cancer. The change in the DNA can potentially lead to cell cycle breaking down and sort of too many cells being made. Okay, so here's our proto-oncogene on the top. Again, don't just look at this and read it over. Look at the diagram and say, okay, what is this? A proto-oncogene. Okay, yeah, we're looking at a chromosome. We're looking at a double-stranded DNA molecule. Not the whole thing, just from there to there. And what does this gene do? The copy. It is the code or the blueprint or the recipe for these proteins down here. Okay? Now, if you take that gene and irradiate it with UVB radiation, or like the folks in Chernobyl, now it's the Ukraine, the high energy rays from that nuclear material get into your cells and they break that gene. They physically break the DNA. When the DNA is transcribed and then it's translated, we form a different protein because the amino acid sequence is now changed, just like with the sickle cell anemia example, right? And so instead of getting these purple round proteins, we get this strange green kidney-shaped protein. And that protein might be better at stimulating growth, okay? So the mutation here leads to a weird shaped protein, and maybe that protein is even better than the original protein at stimulating cell growth. That would be like one of those probably base pair substitutions that we talked about when we talked about mutations. Now, we didn't talk about this type of mutation, but you can have the example where one gene, after it is the cell goes through synthesis, the entire gene can be duplicated once or twice or even 3 times. So here the mutation leads to 3 copies of that gene being present in the new cell. Okay? And if you have 3 recipes or 3 blueprints and the ribosome gets 3 times as many mRNAs, that ribosome may be making 3 times as much of the original protein. And if there's more growth factor, there's going to be more cell division. Okay, so a cell duplication can lead to excessive cell division. And then the one on the right here is a little bit complicated for us, but I guess the point here is there are other parts of the DNA besides the gene that interact with the gene that can lead to that gene being expressed or made into a protein either more slowly or more quickly. Okay, but in all cases, now why is again going back to that question I asked last time? Why do we need a proto-oncogene? Okay, why are these here? Why is this healthy? Because if you're producing the appropriate amount of the growth factor, then you can grow, right? When you're 7 years old and you're growing, that happens because of these purple things are made by the cell when it is supposed to be doing that. Okay? Cells wouldn't divide, bodies could not grow, bodies could not repair cuts, could not repair anything if the cell couldn't divide, and the cell can't divide without these growth factors, and the growth factors wouldn't be there if there weren't proto-oncogenes. It's only when the proto-oncogenes become mutated that they are an issue. Okay. This is another type of oncogene, right? And these are, again, cancer-related genes. These are called tumor suppressor genes. And, right, if you suppress something, what do? You slow it down, you prevent it, if you suppress a fire, then you take that fire and you put water on it, right? A tumor suppressor gene is there as sort of the brake system of the cell cycle control system. So if the cell gets to a checkpoint and the DNA is damaged and the cell does not want to proceed through that checkpoint, it stops at the direction oftentimes of these the product of a tumor suppressor gene, the product being a growth inhibiting protein. Okay, so this tumor suppressor, normal tumor suppressor gene, is transcribed, translated, produces this growth inhibiting protein. When it's there, cell does not divide if it's not supposed to be. Now, if you take that gene and you try to think of something else that causes cancer, say you are exposed to some type of pesticide, then that pesticide could interfere with the copying of the DNA and lead to a mutated copy of that. And then when that mutated copy is transcribed, some wrong codon gets put in there, and then when the mRNA is translated, the protein, the sequence of amino acids is not right. The protein folds up and makes this defective, non-functioning protein that looks kind of like a, I don't know, dinosaur maybe? Like a running dinosaur, yeah. There's his head, his long tail. Could be an alligator this way, where there's his tail and there's his open mouth. Could be some kind of space alien or a slug, maybe, sea slug. Whatever it is, this one sure as hell does not look like this one. This is the shape it's supposed to be. If it's this shape, it will inhibit growth. If it's this shape, it ain't doing nothing, right? Well, it is doing something, so it is doing nothing. It doesn't do its job, and therefore the brake line is cut, cell division is out of control, bam, we have cancer. Okay, so what kind of mutations are the ones that count? Mutations in tumor suppressor genes, mutations in proto-oncogenes. Those are the things that can directly lead to excessive production of cells, right? That can lead to tumors, right? And eventually into malignant tumors, i.e. cancer. Okay. One final note here. This is probably a huge portion of tumors. If you can get that tumor cell, which when they do a biopsy, that's what they do. They take a mass, pull out some of the tissue, they'll put it on a slide. They can look at it and look for signs that it's cancerous. They can get into that cancer cell and sequence the DNA in its nucleus. But it's still a cell. A cancer cell is still a cell. It is one of your cells that is just kind of becomes zombified. And when you look at the DNA, oftentimes a very specific gene, right? The P53 gene is often mutated, right? This is such an important gene that it's really central to many types of cancer. Okay, so let's look at the top, the normal P53 gene. Here's your DNA. Okay, there in the middle we see those bases. Okay, if the DNA during the S phase, or really at any point in the cell cycle, if it becomes damaged, this protein made by another cell probably, or this protein comes along and it's actually kind of rides like a railroad car rides along the DNA string, and it finds that damage. Okay, it stops cell division, so it says, okay, we don't want to keep going here because we've got a damaged chromosome. It can repair the damage, right? It takes those damaged nucleotides and actually cuts them out, and then brings in new nucleotides and seals them in to damage to repair the damage. Okay, if the p53 is able to repair that DNA, then the DNA will proceed through the checkpoint and divide as usual. If p53 does everything it can to repair the DNA molecule and that doesn't work, it is so critical that it doesn't proceed and divide with the damaged DNA that the p53 actually initiates like a self-destruct button on the cell, and the cell will just explode. So if it's damaged beyond repair, p53 says it's not worth it for the future of the whole organism. We're just going to explode this cell. Okay. Now, if p53 is damaged, it's mutated by all those mutagens that we talked about, right? And the DNA becomes damaged in another part of the cell or in a different cell, that damaged cell will be allowed to go through the S phase. That damaged DNA will be copied, right? And so the resulting daughter cells are going to have mutated DNA. Okay. And again, there's a whole bunch of, then at some point, cancer is kind of like a snowball effect. When you get p53, the proofreading protein damaged, then future mutations are less likely to be caught and repaired, right? Because at this point, it's like you've damaged the spell check on the cell, and so that's probably why this is such an important gene to become mutated. And so, yeah. Here, I think probably the big point in this lecture is, combining lectures 2 and 3, combining this idea of mutation and its effect on the final protein, combining this idea of mutation and how it ultimately affects the life of a cell and how a cell functions. And then from lecture 4, we've been talking about cell cycle and how that is controlled. And now, so the two kind of come together. A cell divides appropriately because it's controlled by proteins. If that DNA becomes damaged and the protein is not made correctly, then that protein will not control the cell cycle appropriately. And that's exactly what we're talking about. So all roads to cancer go through these systems here. Okay. So cancer is a cellular disease. It's a disease of genetics. It's a disease of the DNA, right? And this is kind of the nitty-gritty of what we're talking about here. And I think it's important that you guys understand this, because a lot of treatments now for cancer in order to understand the treatments, you really have to understand sort of at least this much about cancer, right? Because we're now finding ways to repair damaged DNA, called cell therapy. And so in the future, I think it's just going to be sort of like standard knowledge that you have to have in order to make it through a oncologist appointment, right? So that's it for this chapter. And we have one more or one more lecture, which I will get posted here soon. And we're going to start to talk about more big picture ideas, less what's going on inside of the cell, and more, how does cancer spread? How do we treat cancer currently? What are some new ways of thinking about treating cancers? And then we're going to talk more about how you can actually inherit cancer, the inheritable component of cancer, because we know that some people are very careful in their life. They really do not take a lot of risks in terms of exposing themselves to mutation, but yet they still get cancer. And whereas other people are reckless seemingly, smoke cigarettes for years and years and years and never get cancer, how is that, right? And yeah, I'm going to kind of bring it all together and finish up this theme in the next lecture. So we'll see you then.